Have you ever wondered what happens following a biopsy? How the pathologist detects the presence of diseases from just a single slide? Well, here's how. After a biopsy, the specimen is colored with a dye. This is for the purpose of better classification of cells in the sample, which, in the next process of whole slide imaging, is digitally scanned and archived for further analysis and annotation. In this video, we're going to compare and analyze two different prominent techniques used to color specimens for observation: immunohistochemistry and immunofluorescence. Immunohistochemistry is a type of immunostaining which involves antibodies to label parts of a tissue sample to make it easier for pathologists to identify. The antibodies are used to detect a target antigen in the tissue sample. The target antigen indicates the presence of a specific disease, and if the antigen is present, the antibody recognizes and attaches itself to the antigen, undergoing a binding process. When the antibody binds to the antigen, the sample stains in a certain color when viewed under a microscope. See the image? The reddish-brown staining shown in the nucleus, cytoplasm, and the membrane indicate the presence of the target protein. This way, the stains allow pathologists to identify the presence of a disease. Immunohistochemistry is mostly used to diagnose cancers such as carcinoma, melanoma, or sarcoma, but also neurological diseases such as Alzheimer's. Its advantages include the fact that it provides long-lasting staining. It can be viewed over long periods of time, unlike immunofluorescence-treated specimens, which are generally short-lasting. It is also very compatible with routine historical samples. It also provides a permanent record of the staining, and in general, it is widely adopted in diagnostic pathology. However, its disadvantages include the fact that it is inefficient for multiplexing. Multiplexing is a technique where we simultaneously detect multiple antigens in a single tissue section, and using multiplexing in immunohistochemistry may result in problems like antibody cross-activity, spectral overlap, and optimization complexity, and better results are usually obtained when applying the technique in an immunofluorescence-treated specimen. Moreover, results of immunohistochemistry are not always accurate. Problems with the antibody may result in false results, such as false negatives or positives. Immunofluorescence is a fluorescence microscope-based technique that uses binding specificity of antibodies and antigens. It is essentially similar to immunohistochemistry, but uses a fluorescent dye for staining so as to obtain a specimen like this. The advantages of immunofluorescence as a technique for specimen staining is that it is preferred for colocalization. This is where two different molecules overlap in a single sample, and that it is easier for multiplexing. It also involves fewer steps. Disadvantages include the fact that labeling can easily fade, and that autoimmunofluorescence, where cells inherently emit fluorescence, can overlap with fluorophore conjugated antibodies and complicate the interpretation of the result, leading to false positives. But most importantly, immunofluorescence as a technique is very expensive.